Okay, good afternoon and welcome to the Simons Electron Microscopy Center's Winter EM course. Today we have Cathy Lawson from Rutgers, from the Department of Chemistry and Chemical Biology. She also wears many hats. She has worked with the PDB and EMDB. This section is a little bit different. This is not covered in any of the Grand Chancellor lectures. We've now moved on to, I guess, emerging fields and new frontiers. Hopefully these topics are interesting. So many of you might be going to the EMDB and looking up structures or depositing them. And Cathy's been involved in a lot of these new initiatives and challenges have been going out because we're trying to establish standards in the field. And what part is a data set, what do you want to archive? So without further ado, Cathy.
Thank you, Ed. So it's a pleasure to be invited to lecture in this course. Thank you so much for inviting me. So I'm going to start talking about the 3 data archives that are available for Cryo-EM work. The 3 I'm showing here, the PDB, the Protein DataBank, the EMDB, EM DataBank, and Empire. And I'm going to go into those in a little bit more detail in the next slides. So the Protein DataBank, it's the single global archive of 3D macromolecular structures, so it's all coordinates. And there's over 125,000 entries, and it was established in 1971. And as you, of course, will know, there's X-ray, NMR, and EM structures in there. The first EM structures were deposited in the 1990s, bacteriorhodopsin, 2D crystal structures. Now that in this year, we now have about 1,500 EM structures and growing fast. Now this organization, it's a very mature archive. It's managed by the Worldwide PDB organization, which is composed of the RCSB PDB in the United States, Protein DataBank in Europe, which is at European Bioinformatics Institute in the UK, and Protein DataBank Japan in Osaka. And actually also the BMRB, the BioMagRes Bank in Wisconsin. So EM DataBank is a younger archive. It's a global archive for maps using any 3D method, and it was established in 2002 at the European Bioinformatics Institute. Since 2007, the RCSB PDB has worked with the PDBE at EBI to co-manage the EM DataBank. We formed the so-called EM DataBank Unified Data Resource Project. And again, this is taking off like hotcakes. We expect to reach 5,000 entries by the end of 2017. Pretty exciting. And the youngest archive of all, Empire, is run exclusively by our partners at PDBE. And it's been available since about 2013. Now this, instead of the final map that would be representing, the structure that you determined, you would put it in EMDB. You could also put raw electron microscopy images into Empire. So the purpose is different. Having this particular archive allows you to make benchmark data available, which has been a difficulty because of the large sizes of the data sets. So emdatabank.org, this is the consortium that I was telling you about. We collaborate with the NCMI group at Baylor College of Medicine. Wachu is the PI of the project, and then we have our group at Rutgers, and then there's a small group at EBI as well. So we have this website, EMDataBank.org, which you're welcome to go to and take a look around. We try to stay relevant to what's going on in 3D-EM. In particular, we provide a portal for deposition and retrieval of maps and atomic coordinates and associated metadata. And we try to provide information about news, events, software tools, data standards, and validation methods. I have been trying for a while to keep up with all the workshops going on, but it's getting harder and harder. It's just a longer and longer list every year. But of course, the 3DM mailing list is a great place to be on if you're interested in that kind of thing. And we have a link to that. So just as I was mentioning, we have quite a bit of growth. This is just in the past 6 years. So we've gone from less than 1,000 entries in 2010 to we're definitely going to reach 5,000 by the end of this year for the maps. And the really exciting thing with the resolution revolution, with all the improved detectors and collecting using movies and improved software for basically led to back in 2010, interesting shapes, but here we are in 2015 with, well, 30 ribosome structures by a certain search. How do you know which one to use? How would you know? And so it's really important to be able to provide some validation statistics. And then here's another reason why it's useful to have good validation. Just as a field, you want to be able to have some kind of mechanism to ensure yourself that you're not leading yourself down the wrong garden path to an incorrect structure, which I think the field has pretty well roundly declared that this particular structure that was published in 2013 is just not correct at all. But you know, they managed to get it published in a pretty high profile journal. It provided an opportunity for a lot of our senior scientists to give some, or to write up their thoughts about how to actually get past this issue of how do you know your structure is right. So with the worldwide PDB, we have a kind of a standard process for trying to get validation going for different areas. So for there was an X-ray validation task force that has met twice, chaired by Randy Reed, and they took their task very seriously. They wanted to provide all sorts of criteria for validation. They actually, all the members of the task force actually worked together and did the calculations and produced a beautiful recommended set of validation criteria to use. And similarly, we've done this for the NMR task force in 3D-EM. There's also been small angle scattering and hybrid methods workshops to talk about validation. So the EM validation task force met in 2010 and resulted in this publication here, headed by Richard Henderson. So Richard Henderson was head of our MAP subcommittee, and Andre Sali was head of our model subcommittee. And next. Couple slides. I'm just going to summarize what was provided in that paper. So for EM maps, This was, I guess, because it's a young field, it should perhaps not be too surprising that they said standards for assessing resolution and accuracy of a map need to be developed. But they also made the statement structural features in a map should be in accordance with the claimed resolution. I'll say a little more about that in the next slide. So for models fitted into EM maps, again, the criteria need to be developed. And there isn't such a thing, if you're familiar with X-ray validation, there's such a thing as real space R factors. It doesn't, I guess people are using correlation coefficients or whatever seems to work, but there really isn't a standard that you could use to compare across, say, different structures. And they also felt, a capability to archive coarse grain representations of models is needed. So in summary, more research and development needed. They did have a few specific things about resolution. So using the Fourier shell correlation curve, which I hope by now you guys know about. I see nods, that's good. So I don't have to explain that. So they said specifically that deposition of a published map should include its full FSC curve to the Nyquist frequency on a linear spatial frequency scale. They also went on to say to discourage using masking because that can significantly change the shape of your FSC curve. You can actually, I guess, cheat and get to a claimed higher resolution than you might otherwise. And fully independent refinement was also encouraged. Now, back in 2010, that wasn't really something that people were doing, and this was actually one of the main, I guess, things that came out of this particular workshop was the idea that the two sets of particles should be separated and never meet again. And so you can use them for validation. Yes?
A quick question about the FSC dots.
Yeah.
What is the significance of the top? I don't know. If you're looking at the top and it's like this plateau before it, slopes downwards, what's the significance of the highest point right before the downward slope? You see what I mean? I've seen people just talk about, oh, look, this is important, but the location of this point apply, and I just kind of never knew why that was.
Well, the higher the correlation is, I guess the point where your curve starts to fall off is you can consider to be the resolution where you're starting to lose information. I don't know if there's other particular importance to that.
But what if it even what if it just started? What if let's say the number of people use it seems kind of arbitrary, so like 0.143 or something like this for the resolution cutoff. What if it has higher resolution as far as that number on the y-axis, but it just started sloping downwards earlier?
Oh, you mean if the shape was not
right, but if it was
the sigmoidal shape?
Slower decrease.
Yeah.
So higher resolution according to this point 143 on the y-axis, but just started, the shape was different. It started sloping downwards earlier, as opposed to kind of a flat plateau.
Well, I do know that there are some cases where you don't see this beautiful sort of sigmoidal shape, and that tends to be for membrane proteins. I've seen like the TRPV1, whenever you look at their FSC, you see kind of two bumps. It doesn't really go off as nicely. And I think that, what was it? Somebody told me a good reason for that. I think it may just have to do with the fact that for membrane proteins you usually have some mass that may not be as well ordered from the membrane. So it could be that, you know, if you're seeing if you're not seeing this nice sigmoidal shape curve, that you're getting, you know, that your map. I mean, I guess under certain circumstances, that's the best you can do. I don't know. I guess I haven't done as many reconstructions myself to know if there's a situation.
Term
Yeah.
Because I view it as self-consistency. I don't really view it as resolution as actually as crystallographers would. But if you are using it in terms of resolution, then whatever the number claims to be, can you get accurate information, whatever that number means to you? So if I say this map goes to 1 angstrom resolution, do you see hydrogen bonding? Can you get that type of information from that? I wouldn't, and that's why we've moved, a lot of people are moving to these ResMap plots, local resolution plots.
That's true.
So we might say resolution, but I think very few people mean it in the same way X-ray crystallographers mean it. And in terms of the shape, that's also reflective of that, because it's probably reflective of your data, but in terms of what number you want to extract out of that, that depends on what your question is.
Mm-hmm.
Because if, let's say I say this is 14 angstrom resolution, but it answers hydrogen bonding questions, then you know how inaccurate that number really is.
Yeah, so just to go back, structural features in a map should be in accordance with the claimed resolution.
Yeah.
And so for that reason, if you can see secondary structure, and if, you know, or if you can see individual residues, then you know you're on the right track, right? You know you have something. You know that map I showed you before, which was very problematic. You couldn't see any secondary structure. But in those cases, there is the recommendation to use some sort of a tilt-based method if you can't see secondary structure, if your resolution is low and you can't do better. So there's various software that will do tilt pair. I guess the problem is actually, I guess, setting up to collect tilt pairs. Does anybody do it here, Ed?
No, but we do collect tilted data.
Yeah.
And
Yeah,
rather conical tilt indirectly, you can do that analysis.
Yeah.
But more people probably should do that validation, but if you're near atomic, you might not. It might not be so crucial.
Right. Exactly. All right. So to address the fact that validation criteria need to be developed, we decided to sponsor challenges. So we've had 2 challenges ongoing, a map challenge and a model challenge. And in both cases, the goals of the challenges have been to develop benchmarks, so basically to pick structures that could be representative for people to look at, to encourage development of best practices, to evolve criteria for validation, and to compare and contrast different approaches. These challenges were developed by expert committees. In fact, the MAP Challenge Committee is chaired by Bridget Carragher, and the Model Challenge Committee is chaired by Paul Adams. And we have, in both cases, a really good groups of committee members who have been very active in helping to develop these challenges. And the hope is that when we're all finished up, we will have some publications for everybody to look at to see the results. So the benchmarks that were chosen, so again, this was 2015 when we started. So all of the MAP challenge targets are available as raw images in Empire. It actually has been an interesting learning experience for me because actually just getting the process of contacting people and saying, hey, would you deposit your raw images in Empire? And well, I guess some of them were already there, but others we had to ask for them to deposit them. And then, you know, depending on the labs, the way the lab did their image data collection, you know, so there were quirks, let's say, that made it a challenge to actually just get the benchmark data put together. But we did finally get that together. For the model challenge targets, that was a little easier because we had the maps at EMDB, so every single one of these is a map that is available in EMDB. The only thing was we went back to the depositor and said, could you give us the half maps, the independent half maps, so that our challengers could use them to help if they wanted to for their modeling. So we finished both challenges, or I should say, or at least the challenge phases of these challenges last year, in 2016, and we managed to get 66 maps total from 27 participants for the map challenge, and 106 models from 16 participants in the model challenge. The difference in statistics tells you how much more difficult it is to do the map reconstruction than it is to do the modeling. And we actually currently have assessments, an assessment phases now in progress for both of these challenges. And if you want, I can say a little bit more about that, but you know, we've basically been inviting people to come in. I think Phil has been participating as well. We would like people to come up with ways to look at, so you know, we have 10 different maps that were all created from the same raw image data. So how could you sort of rank those maps? How would you know which is the best map? Or maybe map A is the best map in this region and map B is the best map in another region. But that's what we hope to learn, and we're going to be having workshops coming up this year for discussions about these assessments. So that's all I wanted to say about that for now. And then the last thing I wanted to cover was deposition. So, some of you may be doing structure determinations, and so I'll just give you a little overview of how you actually go about depositing structures into EMDB and PDB. It's actually really nice now, at least that's my impression. When we started in 2007, we had separate systems for depositing maps and depositing models. So you would go to EMDep, deposit your map, and then we invented a cute little thing where you could push a button and at the end of your map deposition, and if all went well, your metadata would be transferred to PDB, to the PDB deposition system, and then you know, so your microscope model, you know, your microscope voltage, sample information, so how you made your specimen, those kinds of things would get transferred over to PDB. But that was sort of a hodgepodge temporary solution, and what we really wanted was a system that could just handle map and model deposition at the same time. And so as of January 2016, we've had that. And it also, so this one-dep system also collects structures from X-ray and NMR, as well as EM. So you can choose to deposit a map, or use to deposit a model. You can use it to deposit a map and a model. And a validation report is provided. At this point, as I will show you, the validation report for EM is kind of simple. But when you come to the deposition interface, the first page, it asks you what experimental method you're going to use. And if you pick electron microscopy single particle, as most people will do, but you can also choose helical, subtomogram averaging, or tomography. And if you choose those other methods, the questions you get asked are a little bit different. They're tailored to the different methods. So for instance, if it's helical, you'll be asked your sample molecular weight per unit length instead of just your molecular weight. So then you get asked a few questions. Are you depositing coordinates with the submission? And so you can say yes. If you happen to say no, it will actually ask you, oh, wait a minute. Sorry, it's the next one. So it has the associated map and deposited previously. So if you say yes here, it will ask you a few additional questions because it's actually now required to have a map associated with a PDB deposition. And so if you say yes, then you can it will ask you questions that will allow you to link it to a previous deposition of a map. And thank goodness for these questions because before we used to just ask requested accession codes and ask people to check boxes, and I was always worried nobody's going to know what to do here. So so these then get automatically filled depending on the questions you or how you answer the questions. So the file uploads. We have, so for 3DM, it's actually kind of more complex than it is for crystallography. So after they set up one dep for crystallography, because you just basically have coordinates and you have structure factors and that's it. For EM, because in the EMDB, we actually allow a lot of extra files in addition to just the map. So just to go through it, so the coordinates you can upload in either of these two formats. Those are going to end up in PDB, of course. You have to deposit a map, and it has to be in MRC or CCP4 format. You need to upload an image for EMDB, and that's actually a good thing because annotators can make images of your maps, but they're not going to be as pretty. So if you want a pretty picture of your map in EMDB, then make sure you upload a nice image. You are allowed to have additional maps. Now this is kind of an interesting point because we get questions frequently. I used, from a single set of image data, I produced 20 maps because I, have all these different conformations of my molecule. And what do I do? Do I need to upload, do I need to make 20 different depositions? And the answer is maybe. If you want an EMDB ID associated with each one of those maps, then yes. If you want, if you just basically have one representative map that you want to have associated with an EMDB ID, then you can just upload all of your additional maps here. And you can just keep uploading as many as you want. You're allowed to upload masks, and then the path maps are actually a separate type of upload. Now, so we know specifically if we're getting your half maps that could be used for FSC calculation. And structure factors can be uploaded. That's kind of an interesting one. So for, of course, for 2D crystal structures, you expect to have structure factors, but we actually also get structure factors from people who take their map and invert them into structure factors for refinement purposes. PDB doesn't really want them because they're not primary data, they're derived data, but EMDB will take them. And in addition, you can upload an FSC file, which I'll say a little bit more about. If you have a ligand, you can upload an image. PDB actually spends, or I guess historically, for PDB annotation, you know, we have a huge group of people who do nothing but preview the data that comes in and try to make sure it's as accurate as possible. There's a whole process developed for ligands, which maybe wasn't so interesting for EM people 5 years ago, but should maybe be more interesting now because, you know, you can probably see ligands in your maps. If you have something that's unusual, you can provide an image for it, and that will help them to identify whether it's something that's been seen before, in which case it will be assigned an ID, a ligand ID code that will allow you during search to find all entries that have that same ligand, which is pretty awesome actually. So this is the, so there's a server at PDBE, pdbe.org/FSC to upload 2 maps to create an FSC curve. It produces this nice image that you can download, and it also produces an XML file. And so the XML file is basically just an XY data points of this curve. And it also provides you with all the different possible criteria you might want to use to define your resolution. So if you use the server, or actually if you use there's those software packages that also produce this XML curve, you can upload that curve as well. Now, one benefit of doing that is eventually these are going to start appearing on validation reports. So I think, yes, so that's my next slide. So currently, the OneDep system produces a very simple validation report for EM structures. It's only for EM models, and it only gives information about the quality of the model, but not the model with respect to its fit to the map. And there's really very little about the map, or actually there really isn't anything about the map. So we have kind of a table 1, just with some basic information about how the data were collected. And then there's this bar chart here, which provides information about the quality of the model. So clash score comes from multiprobity. And so if you have, you know, so the higher the number, the worse your model is. Now, and then you're also given these bars here. So the black bar is the percentile relative to all structures in the archive. So in the PDB archives, that includes X-ray and NMR. So, and then we also provide percentile relative to all EM structures. So this particular structure fares a little better relative to EM structures. Now, for X-ray structures, because there's so many of them, they do percentiles relative to X-ray structures in fine resolution bins. Okay. I see the heads nodding, so you're familiar with this. But there aren't really enough EM structures to be able to do anything like that. So in the future, probably that will change. So this particular structure did pretty well with their side chain outliers. Pretty good. And so we're going to hopefully, so we soon add some additional information to the validation report. And I hope maybe this will work. I'm going to see if I can just go to this. Oh, can I make it big? Oh, okay. So I don't know if you're familiar with the PDBE's visual analysis pages. So basically we're planning to incorporate what you see on these pages in these reports. Oh, I should have mentioned, here's something also really important. So journals are starting to request these reports. When you submit a manuscript, don't be surprised if they say, hey, please also include the validation report so that we can send it to reviewers. And so having these kinds of things available for reviewers could be very useful. So among the things that will be included are these projections. And I think it's actually pretty useful to be able to see. I mean, you can tell very clearly here, this is not a masked structure. There's no masking in this volume. Central slice, orthogonal surface views. Of course, that has to be done at a particular contour level. We do ask for people to provide their recommended contour level. It's not, there isn't one specific, we don't really have guidance on the contour level other than, I guess it's, the one that you would use to view your map the most. What gives you the most information. And then in addition, there are these graphs that are produced. So you have the map density distribution. So that just gives you the distribution of density points for the map. There's a volume estimate graph. So here in red is the recommended contour level. The sample is here. So in this particular case, they recommended a contour level that's maybe a little higher than you would actually want if you wanted to cover all the sample volume. But of course, that doesn't exclude the possibility that you're actually, as you come down in contour level, you might be adding noise. So that, so again, this red was what the depositor told us was a reasonable contour level. And in this case, they did provide their FSC curve, so you see it here. And then there's a rotationally average power spectrum. So in reciprocal space, you can get an idea of what's happening here. And obviously, there's been some kind of filtering going on at this point where things have come off. And so this gives you some basic information about the map, and we're going to be including those in the validation reports. Oh, so in addition, we have this map model fit. It's basically by eye, so you can see the model superimposed on the map. Right now, in the annotators that I work with, this is what they actually, when they get the map and the model, they just bring them up in Chimera to see if they're fitting. They just look visually. Creating these images actually will make things a little easier for them. So they'll be kind of nice And then there's a atom inclusion at the recommended contour level. So you get a little plot of whether atoms or residues are included within the map. And I think, yeah. So head back. Okay. So that was pretty much it. As I mentioned, our project team, Hua Chu, our PI at Baylor, actually he'll be moving to Stanford this summer. And I work with Helen Berman at Rutgers University, and with Gerard Klebeck and Arden Patler done at Protein DataBank in Europe. And I'm done. I will happily take questions. Anyone?
Have any of you used EMDD to download anything, view anything?
I've pulled lots of maps. Yeah.
Yeah.
I guess lots.
Lots?
A handful.
Handful? Do you know if they're any good?
They seem to be good. So the maps I've downloaded, they also had, like recommended contour levels, which was really helpful to like I found that key to actually using the maps that I downloaded to see what the builder saw.
Right.
To sort of decide whether or not you agree with what
Have
they
you ever
did.
thresholded at that contour level, remove the PDB that's associated with that, and would you be able to say, I could confidently build within that volume? Because that's a big question right now, because a lot of people want to model build these EM maps. And
Yeah.
let's say you don't have any methionines that you can orient yourself.
Yeah. So, I mean,
A
I've
lot of
definitely
loops.
looked at a map where there was clearly like, oh, there's a piece of density here they didn't build into, and it's probably beta strand, but like, did I want to do it? Not really. That's a lot of work, right?
Right.
It's like I could certainly try, but I don't. Yeah, I did not actually participate in the challenge because I mean, it's building by hand into the maps is hard.
Right.
Yeah, it is hard to do.
Yeah.
Yeah, it was interesting. It's a really different community too between the people who participated in the map challenge and the people who participated in the model challenge. I guess for the model challenge, there were a lot of people who are developed software that was specific for crystallographic use, and they're modifying it to make it useful for Cryo-EM work. All right, anything else? Okay, well, thank you.
Quite simple. Let's thank Cathy. And next week will be validation methods by Tom Waltz. It's going to be a very big open-ended course or class, because we're still trying to develop validation methods, or at least ones that we can all routinely do.
I think I'm going to have to listen in.
Right.
Online.
Yeah.
I mean, it's easy to say to do certain experiments, but to do it routinely, we might not necessarily be there. I mean, you can understand, you're at the end of a 2-day data collection, it's like, oh, by the way, you should do another half day of this validation method. It might be a little bit hard.
Yeah. Okay.
Sorry.
All right.
Okay,
Great.
thank
Thanks,
you.
everyone.
Thank you.
My feet have finally cooled down.
Are you the only one from Columbia taking it for credit?
No, Mike is as well.
Okay, so you too.
Yeah.
Do you know is Joachim the course registrant?
Yeah, you're in charge.